Hi guys, Dr. Orlando here with a video on enzymes. First thing we're going to talk about is what is an enzyme. So an enzyme is a protein. And so it has a specific shape, very important. Remember with proteins, you always have to remember that those primary, secondary, tertiary, and quaternary structures of proteins are what make that protein special. What makes that protein have a specific shape, and it gives it its specific function. So the shape determines function. So if it's the right shape, it works well. Okay. All right. So what is its function? Its function is to be a biological catalyst. That means it speeds up chemical reactions in the body. Okay, how does that do that? Well, it has two things that it can basically do. It can either do a catabolic reaction. In a catabolic reaction, the enzyme is breaking down something. And in a catabolic reaction, a generic one might look like this. If you're breaking down, you'll take something that is it's a little unit there, and the enzyme's job, the enzyme there, over the arrow, remember? And it will break it down into its purple, into components. That's catabolism. Okay. So you started with this big thing of A, B, and it's broken down. The enzyme breaks it down into parts A and part B. Or you can have an anabolic reaction, or an anabolic enzyme. Okay. And with an anabolic enzyme, you're building something. So you would have A plus B is going to yield AB. Put our enzyme over the arrow. Do our circles around these so you can see them separately. So we built AB. Okay, and remember this. Remember that enzymes often have the last name ACE. So you can write EACE in here. EACE. Enzyme ACE. To help us remember that enzymes have the last name ACE. Okay, so now we're going to talk about enzyme anatomy, and I just want to show you our key players. You're going to see these throughout the rest of this video here. So the blue Pac-Man looking guy is our enzyme. Remember, he's a protein. He's folded up. His amino acids are folded up to make that shape. And you can see I've got two arrows pointing at our enzyme. So let's go ahead and label these. This spot that looks like the Pac-Man's mouth is the active site. The active site is where the substrate attaches. And then this other one over here is the other site, also called the allosteric site. Okay, meaning other shape. Okay, and allosteric site is also referred to as the regulatory site. When we talk about controlling enzymes, we can use this site to help control this enzyme to make it work better or to inhibit it. Okay. All right, so these guys here, so this is, let me label everybody, enzyme. These two little triangles with the letter red triangle and yellow triangle with the letter S, these are our substrate. Okay. also the reactant. And then when we put them together, we have product. And what was made when these guys are together, we call it the enzyme substrate complex. Okay, now imagine you have a beaker full of substrate, the little red triangles and the yellow triangles. And you're hoping that these will form some kind of product, the product of the red and the yellow triangles hooked together. That's what you're hoping for. Will that ever happen in here? It could. Randomly, the one could bump into another one in just the right way that, that they bind. It could happen. It would be extremely slow, however. It would be very slow. Because it would be random, and because the substrates actually repel one another. Remember, these substrates are made up of atoms, and atoms have electrons floating around them, and those electrons are negative. And the electrons from one atom actually repel the electrons from another atom. The clouds bump into each other, and you get this bouncing off happening. So they want to combine, right? But they aren't able to without an extra push. And that extra push is given, that extra push that's needed is activation energy. The energy needed to start a reaction. So activation energy, the energy that's needed to start a reaction, the energy that's needed to overcome those electron clouds repelling each other, and the energy that is provided by enzymes. So activation energy needs to be provided by enzymes. Okay, so I added enzyme to this beaker, and you can see that some of the substrate and enzymes are still repelling themselves, but guess what else is happening? Some of these enzymes and substrates, some of these substrates, excuse me, are going to be attracted to the active site of the enzyme. They have the active site of the enzyme has amino acids there that are attractive to the substrates. They want to go there. It's a happy place for them, and it puts the substrates in the right orientation so that they can bind and that the electron clouds are no longer a problem. Okay? So it brings them in, puts them together just like this, and then they are released. Released just like this. So they come out, and there they are. Now you've got a product made. Okay, now you might ask, how can you increase the rate of this reaction to make it go even faster? Well, the rate of the reaction is dependent on a few things, one of which we can point out here. Notice I have only 2 enzymes in my beaker. It would take a while for these 2 enzymes to put all those substrates together. So if I added more enzyme, so if I increase the concentration of enzyme, these will be things that will increase the speed of the reaction, increase reaction rate. So if I increase the concentration of enzyme, that's going to speed it up, because now you've got more of these Pac-Mans to put these guys together. Okay, then I can increase the amount of substrate. And you might say, well, how does that work? Well, that's to a degree. You can increase substrate so that that time in between when this product leaves and others find their way to it, has that time in between, that lag time. So the more substrate you have in there, the less time there will be any free enzyme floating around. It's kind of like having a waiting line or a waiting list to get into the enzyme if you have a lot of substrate in there. That will hit a limit. When all the enzymes are saturated and you can't fill them up anymore, adding extra substrate won't help. So increasing substrate to a point. Okay. So both of those will increase the reaction rate. Okay, so now we're going to talk about how to control these enzymes. We need to make sure that these enzymes are doing what they're supposed to do, when they're supposed to do it, and where they're supposed to do it. So the first thing that we can do is we can put them in a vesicle. Remember, these enzymes are proteins. So how they are made, you take the DNA from the nucleus, and DNA is used to make RNA. That RNA, remember, is the recipe. It goes to the ribosome, and the ribosome is the workbench where we make the protein. And it could be on the ER, could end up in the endoplasmic reticulum, could go through to the Golgi, where it could be packaged into a vesicle. Now, this enzyme could be moved to the edge of the cell, where it can be released by exocytosis, and therefore the enzyme only works outside of the cell. So it kind of puts it in a cage. So imagine if this enzyme were a protease, an enzyme that breaks down protein. We wouldn't want to just release him inside of the cell, because if we did, he'd start going around munching up all the cell's proteins. Well, he's not supposed to. He's supposed to munch up proteins that are outside of the cell. So we put him in a little cage, a vesicle, which is a small membrane like sac, and it can be moved for exocytosis. All right, number 2, we could take that enzyme and we could make sure that he is membrane bound. Membrane bound. What does that mean? That means after he is made, DNA to RNA, RNA to protein on the ribosomes, after he's made, he's shipped to the membrane. This is what I'm drawing here is our phospholipid bilayer. This could be the plasma membrane. It could be part of the internal membrane structures, wherever this enzyme is needed. But he's membrane bound. That means his location is controlled. He can only do his job right there. He can't go anywhere else in the cell and do it. He's only going to work in that membrane. All right, and then last but not least, actually not last, second to last, we can secrete them in an inactive form. Inactive form. So here I've drawn, I'm going to move him up, zoom in a little bit. So here I've drawn him so that you can see his Pac-Man mouth is filled with something, right? So right now, he would be saying, my mouth's full, I can't work on anything. So the active site is occupied at this moment. Okay. Or there could be other ways to inactivate an enzyme. Whatever the cause is, it is inactive. So even if it is in the cell floating around, he's inactive, so he can't do anything. And the only way to activate him, well, there are ways to activate him. Sometimes they're activated by other enzymes. Sometimes they're activated by, say, hydrochloric acid. Okay, whatever the reason is, they become activated. So this little pink blocker would be removed. Let's say that this is in the stomach and it's hydrochloric acid that's floating around. It's the hydrochloric acid that goes in there and removes that chunk. And now this active site is ready for business. It's ready and waiting. Okay, so that is another way that we can control enzymes. We secrete them in an inactive form, and they're not activated until they are in the location that they need to be. In this case, with the hydrochloric acid, they're activated once they're in the stomach. Okay. All right, so those three are ways that we can control enzyme. The fourth way, it's going to take a whole other page here, number 4. This is inhibition. So we'll move on to the next slide. So with inhibition, there's different types of enzyme inhibition that we're going to talk about. Just to review our enzyme characters here in the story here, we've got our enzyme, we've got the substrates. This is our enzyme substrate complex, and that's the product. Okay? So enzyme substrate complex, and product. So the first type of inhibition, how we're going to stop this enzyme from working, is called competitive. And in competitive inhibition, the inhibitor will compete for the active site. So it also has affinity for the active site, attraction for the active site. However, it can't be worked upon by the enzyme, and it just sits there and blocks. So sometimes you might call these blocker drugs. They block them. Okay, so here in my picture, I've got this little green blob. This is our inhibitor. Sometimes inhibitors are the same shape as the substrate, so they fit there so perfectly. Sometimes they're just close enough to get in there and block them. Okay, so let's move this guy over to block the enzyme, and then these poor little substrates, they cannot get in. Okay? So it's a competitive inhibition because the inhibitor competed for the active site. Okay, next one. So the next one is non-competitive inhibition. And again, we're inhibiting the enzyme from doing its job. But the non-competitive means that the inhibitor is not competing for the active site, and instead it attaches at that regulatory site for the allosteric site. So sometimes we will call these allosteric inhibitors. Okay. All right, so let's see how that looks like in our drawing here. So this is our non-competitive inhibitor. It's going to attach to the allosteric site, and when it does do that, it closes the mouth. In this case, this is just the example I'm doing. It changes the shape of the active site. Okay, it always changes the shape of the active site, but in my drawing I showed it as the mouth is closed on our enzyme. The active site is just not available at all. Okay, so now these substrate that want to get in cannot get in because the active site has changed changed shape. Okay, so they can't get in. In this case, I drew it as being closed. So that's non-competitive inhibition. The enzyme is inhibited, true. It is non-competitive because the inhibitor binds at the allosteric site instead of binding at the active site. The third type of inhibition is called negative feedback. And with this one, we're really controlling a pathway more than controlling one particular enzyme. So this type of inhibition could either be competitive or non-competitive. But what I want you to understand about negative feedback is you're looking at a big picture. You're looking at an entire metabolic pathway that's being inhibited. So how does that happen? Well, here's a metabolic pathway that I've drawn. All my little colored boxes are substrates that are being worked on the enzymes. Okay, the little letter E's over top of the arrows. Okay, and so if we keep feeding this pathway with the initial reactant, right? And the first part is the reactant, the ones in between are the intermediates, and the last thing produced is the product. So if we keep feeding that red block substrate into this reaction, we will keep getting product, right? And that product will accumulate. So let's make a bunch of product accumulating over here. Well, you're really feeding this pathway, making lots and lots of product. Over time, some of that product may feed back, may diffuse, or however it gets there, feed back to one of the enzymes in the pathway. We'll make it go to the first enzyme in the pathway. And when it goes there, it shuts that enzyme down. Okay? It shuts the enzyme down. When you shut that enzyme down, what happens to production? Production stops. Right? So then maybe this product begins to get used for whatever its purpose is. Then this guy can diffuse away from the enzyme, and the pathway can continue on again. So we call it negative feedback because the product of the reaction feeds back and shuts down production. So the product of the reaction inhibits an enzyme in the pathway. So it's self-regulating so that you don't overaccumulate too many of those blue substrate blocks. It shuts it down. That's negative feedback. All right, last thing I'm going to talk about here then on enzymes is their environment and what makes them happy and unhappy. All right, so remember enzymes are a protein, so they have a three-dimensional shape, and that is very important that you maintain that three-dimensional shape because that active site has to be the right shape for the substrate. If you don't have the right shape because it has been affected in some way, if it changed its shape, we say that the protein has been denatured. Like the example I gave in class, if you take the white of an egg, which is protein, albumin, and you cook it, it turns white. So now you can't uncook that egg. You've changed that protein shape, and now it is white instead of clear. It has been denatured. Okay? So a couple things we talked about in class that can affect how environment affects enzymes. We talked about pH. Okay, so there's no one pH that's perfect for every enzyme out there, but for each enzyme, they have a pH range that they work at. So they have a specific range. So it depends on what kind of enzyme you are, right? If you are in a cell, the cell's pH is in balance with our blood pH. So in a cell, you're going to have around a 7.4 pH where those enzymes are working well. If you're an enzyme that works in the stomach, stomach acid is about 2. So those enzymes like to work at a pH of 2. From the stomach, food that's processed in the stomach goes to the small intestines. And in the small intestine, right, the first part of the small intestine, the pH is about 8. So can you imagine these enzymes that were over here in the stomach doing their job, chopping up those proteins, and they get to the stomach, or go from the stomach to the intestines. And they went from a pH of 2 where they were happy to a pH of 8. They're very unhappy. So those enzymes that worked in the stomach don't work in the intestines. Then you got to add in new enzymes for the intestines. Okay, so they all have a specific range where they work right. Why is that? Well, when the pH is appropriate for them, they are folded properly. If you change the pH, you're probably changing the hydrogen ion concentration. You are changing the hydrogen ion concentration, and that'll cause the hydrogen bonds that are holding that three-dimensional shape to change, and then the shape changes. All right, temperature. All right, again, it depends on what the enzyme is. Each enzyme will have a temperature that it likes to work at. If we're going to talk about humans, animals in general, warm-blooded animals, we would say that if the temperature goes down, a decrease in temperature, you would likely see a decrease in enzyme activity. If you go to the worst possible scenario with decrease in temperature, if something is frozen, the molecules can't move around, and if they can't move around, they can't be worked upon by an enzyme, and so the enzyme can't do its job. So imagine going from very warm to getting colder and colder and colder, and the enzymes are just slowing down as it gets colder and colder. If the temperature is very high, let's go with slight. Like a person has a slight fever, so a slight increase in temperature, that actually gives an increase in enzyme activity. It's like turning the temperature up on the stove just a little bit. It just increases the action ever so slightly. They can work a little bit faster because the molecules that the enzyme are working on are moving around more quickly. As you add heat to them, they move. Okay? So it just increases the enzyme activity slightly. Now what if you have a big change in temperature, like let's say we are going to boil or cook something like an egg, that will significantly decrease enzyme activity. So if you have a big fever, 105, 107 up there, now you're looking at the danger zone of denaturing somebody's enzymes because they're literally cooking them in their body. Okay, and that would be bad, not consistent with life. And then normal temperature, you would expect normal enzyme activity. Okay. Last one I talked about in class was salt. Normal salinity of the blood, 0.9%. That's where sodium chloride. That's where our enzymes like to be. And if salt should increase, this isn't, let's talk about a significant increase in salt that we've done to something. Let's say we're going to take some cucumbers and we're going to pickle them. So in the jar we're going to put vinegar and salt and maybe some sugar. We're going to put a lot of salt in there, a lot more salt than is actually inside of the cells. So that salt will denature enzymes as well. So increased salt will decrease enzyme activity. Okay. And that is the point of pickling something. Well, that is the reason that you add salt to it, because you want any bacteria or critters that are in the cucumber, surface of the cucumber in the jar to die from the salt and the vinegar that is there. The pH and the salt together will destroy all those enzymes, and then that critter will die and won't cause the food to spoil. All right, so that is enzymes. There were a lot of examples we talked about in class, and there are more details in your notes in class. So make sure that you go through them and make sure that this makes a lot of sense to you. If it doesn't, make sure to come to review session, give me a call, or email me. Thanks. Have a great day.